secretions of enzymes. And there are many. They're usually, the majority of them are actually inactive precursors. This is very important for the pancreatic gland, that they are secreted, produced, synthesized, and produced and stored as inactive precursors to the active forms, the majority of them. I will point out which are active forms. To maintain this inactivity, we have secreted along with the inactive forms an antitrypsin, and you'll learn soon why how important this antitrypsin is in with the other secretions. It tries to keep the enzymes in an inactive state, and if it becomes overwhelmed, we can get pancreatitis occurring. Okay? So those are the enzymes, and we show you how they're activated shortly. All right, so we have to go to mechanism of secretion of this large volume of electrolytes, which is rich in bicarbonate. And it's an active process, okay, of secreting bicarbonates. And this is what is mainly accepted. All of it is not worked out, but we generally accept, based on the investigation, that bicarbonates are the actively secreted ions. Okay? And as usual, that is largely due to the sodium gradient that is created in the cell because of the pumping action of the sodium out of the cell, creating that gradient in the cell. The majority of the bicarbonate, and you can understand that we have hydrogen ions in the stomach, in the parietal cell. We need a lot of bicarbonates to concentrate and secrete out into the lumen of the ducts, central acinar and ductile cells. A lot of bicarbonate. And that the majority of that bicarbonate is going to come from the blood. So that bicarbonate will have to cross the basolateral membrane. And it does so because of the sodium gradient that is created, providing the energy, and it is actually co-transported along with sodium into the cell. So if you look at the basal aspect, which is the area closer to the blood, okay, you will see that sodium co-transport with your bicarbonates, okay? So lots of the bicarbonates actually enter, mouse, use the mouse instead of the pointer. I can't see the mouse very well. All right, so we have right at the top there, the co-transporter of your sodium and bicarbonate. In addition to that, we do get CO2 entering the cell, combining with water to form a weak acid, your carbonic acid, due to the activity of carbonic anhydrase. And because it's a weak acid, it's always in association with its components. So we get bicarbonates also from CO2 entering the cell to add to the large amounts of bicarbonates which are coming from the blood into the cell. All right, so we have that additional factor which creates bicarbonates inside the cell. So some people may wonder, well, why does it require energy to transport bicarbonate now that is being created, accumulating in the cell into the lumen? And that is because the lumen is electronegative. So we have to transport and concentrate a negative ion into an environment that is already electronegative. The electropotential across the membrane, the epithelium of the duct cells there is negative in the lumen with respect to the inside. So we need energy to concentrate. So movement of the bicarbonates against both the concentration and electrical gradient. Energy is provided, and that comes from the pumping of sodium out, creating the sodium gradient for the movement of sodium into the cell. And that is done so with bicarbonate being transported. So that carrier transports the sodium and bicarbonate into the cell, along with the bicarbonates being created inside the cell. So we have lots of bicarbonates now into the cell. And the hydrogen that is being created is actually being exchanged for sodium as well. So both the hydrogen sodium exchange as well as the co-transport of bicarbonate with sodium is due to the sodium gradient that is being created due to the pumping action of the ATPase. Okay? So that is as simple as we can get it. So we have the bicarbonates and we have to get it into the lumen. Well, it is done because of the presence in that luminal aspect or the apical aspect of the cell. If you look closely at the diagram, you will see a channel which transports your chloride and your bicarbonate. In order for bicarbonate to be secreted into the lumen, we must have luminal chloride first of all. Okay. We must have chloride in the lumen. And that is dependent on this same, I mentioned it before, chloride channels, and one of them that actually transports the chloride and bicarbonate is the cystic fibrosis membrane transporter regulator that we have referred to already. And this is a very important channel as dependent that allows chloride ions to move into the lumen and then for that to be exchanged again with bicarbonate. So you see the chloride movement in, and then you see, of course, the movement of your bicarbonates into the lumen. Right? You need that exchange to take place. And if this channel, the cystic fibrosis transmembrane regulator, is not working, is defective, has not been synthesized, has not been put into the apical membrane of the duct cells, you're going to have a problem. And this is a result of a condition called cystic fibrosis. So cystic fibrosis affects many cells that require chloride transport, and this is a mechanism that requires your cystic fibrosis transmembrane regulator channels. That is dependent, the opening of that channel is dependent on the creation of that second messenger, cyclic AMP. So you need to increase cyclic AMP levels in order to open these chloride channels. And that is dependent on secretin release. Secretin. And secretin release is dependent on acid. So acidity of the intestine will cause the release of secretin. And if you recall, I told you to look up secretin and gastrin and these things. Secretin is released from S cells under the influence of acid. The secretin is the stimulus to cause these channels to open to allow the chloride factor, the chloride movement, so that bicarbonates can be secreted into the lumen. So we have all of the bicarbonate that is accumulated in the cell to actually move and be secreted out into the lumen. What the diagram also shows is that sodium will move paracellularly, and that of course will encourage the movement of water across the cell into the lumen. Okay? Because of the osmotic gradient created, that sodium chloride, salt, that sodium, which always tends to cause water. It pulls water with it. It's a small ion that pulls a lot of water with it. So once it moves into the lumen, it is going to pull water with it. And as a result of that, you get an isotonic secretion, which is full of bicarbonates. But pay attention to that cystic fibrosis transmembrane regulator chloride channel, because that is ultimately important for the function of the pancreatic gland in terms of its exocrine secretions. Cystic fibrosis is a problem. Okay? All right. In the intestines, you essentially actually will get this result where the acid from the stomach will combine with that bicarbonate to create a weaker acid and some salt. So you're trying to sort of neutralize the area and of course increase the pH from the very low pH of the acidic juice. Now, sodium in the large, so I already said all of that. Okay, sodium moves paracellularly. Water will move to accommodate the osmotic gradient, and you have the resultant isotonic exocrine secretion from the pancreas, which is able now to neutralize the lumen of the intestines and especially the duodenum, and allow the enzymes that are also going to be secreted along with this isotonic secretion to carry out their job effectively. Now, in the larger ducts, it is felt that the bicarbonate that are secreted out can be exchanged with chloride. As we had indicated for the saliva glands, it is felt that some of the bicarbonates, not all, some of the bicarbonates are actually reabsorbed in exchange for chloride ions. And so there is a somewhat of a chloride bicarbonate inverse relationship that is seen when we do the experiments. Okay? So secretory rate is going to affect the amounts of bicarbonates that are actually secreted out. Usually when there's a stimulus, such as food, causing the release of various substances, in particular hormones, humoral factors, you're going to get increased rate of secretion, which is going to most likely increase your bicarbonate content. Now there are two component theories that try to explain this, and maybe both actually contribute to how this change in composition is affected by secretory rate. And one of the component theories says that the acinar cells, the acini, secrete a very low volume of sodium. Remember the central acinar cells, a low volume of sodium chloride. So the main ions secreted by the acini are sodium and chloride. And the duct cells, upon stimulation, will secrete lots of bicarbonates. Okay? In response to stimulation. So they secrete bicarbonate, but in response, lots of bicarbonate in response to that stimulus. The other component theory says, look, the duct cells simply secrete bicarbonates. They just secrete bicarbonate as their main ion. Okay? And in the large ducts, that bicarbonate is exchanged for chloride ions. That is what another group proposed, that that is what they feel largely. So the cells really, the duct cells really secrete bicarbonate, which is exchanged in the large ducts for chloride. So when we look at and you see that inverse relationship. So if you look at whether it's low secretory rate or high secretory rate, both component theories contribute, and both component theories can be right. Because at low secretory rate, if you say that the acinar cells secreted sodium chloride, namely low volume, and the duct cells will secrete bicarbonate if there is stimulus, then if there's no stimulus in low secretion, then chloride ions will predominate. So it works out for that theory. The other theory is that the cells simply secrete bicarbonate. So at low secrete, and if they secrete bicarbonate in the large ducts, they're exchanged for chloride ions. So you can just understand that at low secretory rate, as we explained with the salivon, there's lots of time for exchange with chloride ions in the ducts. And so the end result is that you're going to get predominantly chloride ions with sodium. Question? energy is a secondary energy, secondary active transport. That energy is provided by the pumping action of the ATPase, the sodium potassium ATPase pump. You know about primary and secondary, right? So the energy to drive, the energy is by that pump creating the gradient, which allows sodium to simply diffuse in. Yeah. So the chloride, the bicarbonate, not the chloride, the bicarbonate ions require, if you block that pump with warbane or so, you will not get bicarbonate secretion. But bicarbonate secretion in the lumen is dependent on chloride exchange.
So how do you get that inverse relation based on these two principles?
What I just explained, right? Chloride movement, bicarbonate exchange requires the cystic fibrosis channel, right? What we are saying, so normally you get bicarbonate secreted in the lumen. As an isotonic secretion is passing down the ducts, one of the theories says that in the larger ducts, there's some evidence that some of that bicarbonate is going to be reabsorbed in the ducts for chloride. So there is a bicarbonate exchanger in the ducts. I haven't shown you that. I'm just stating it. So the component theory that says there is that exchange, it means that at low secretory rate, there's a lot of time for that exchange to occur in the large ducts. You can appreciate that, right? Similar to what we talked about in salivary glands. Lots of time for the exchange. So more bicarbonates are going to be exchanged for chloride ions in the large ducts. And so the final secretion that comes out will be a secretion which is richer in chloride than in bicarbonate at low secretory rate, according to that theory. The large ducts. That
was what the second theory.
Yes. And the other theory is that it's a small secretion that the acinar cells produce of sodium chloride. There's not much stimulation for bicarbonate secretion by the ducts, and so chloride and sodium predominate. And just the opposite for high secretory rates, where the sodium chloride that is being secreted by the acinar cells is going to be overwhelmed by the stimulatory secretion of bicarbonates by the duct cells. So bicarbonates will predominate, and so that will be the main ion that you see. That theory. The other theory is that the bicarbonates are the ions that are being secreted by the duct cells, and here is stimulation to create high secretory rate. There's not going to be enough time to exchange at the large ducts, and therefore bicarbonates will predominate. So both theories result in the same end result, and both theories may contribute to the secretory rate in terms of the bicarbonate secretion. So at high secretory rate, you expect sodium bicarbonate to be the predominant ions in that isotonic secretion. All right? So just go over, just think about it. So this is just to show you the difference, comparing the two, the salivary gland, when secretory rate goes up in the salivon compared to the pancreatic secretion. And just to point out that sodium is always high in pancreatic secretion, similar to plasma. Potassium is similar to blood plasma in the pancreatic secretion. Is the bicarbonate chloride that changes, as inverse relationship, and compare it with the salivary glands. Okay, and we'll go back over that. But you can look at it when I upload the slide for you. All right, so another component that of importance are the proteinaceous component of the pancreatic exocrine secretion. So let me talk a little bit about the mechanism of secretion. And I left mechanism here of protein secretion, but generally speaking, even in the peptic, the chief cells of the stomach and other areas where protein is being synthesized and secreted, it's a similar sort of mechanism for enzyme secretion. So it begins with the exportable proteins. Exportable proteins, and these are associated with polysomes, which are part of, they're attached to the cisternae of the rough endoplasmic reticulum. So we need our rough endoplasmic reticulum. Of course, we need our nucleus, because that has to dictate how the proteins are going to be synthesized and all of that. Of course, this is cellular physiology, cellular things. So I'm not going to go through that. You should know about all of that stuff already. But we require the rough endoplasmic reticulum as well as the Golgi apparatus. So it starts there with these exportable proteins in association with polysomes at the cisternae. And what happens is that as the amino acids are being synthesized and attached and elongated, that protein is being elongated, it is carried by these leader amino acids into the cisternae of the rough endoplasmic reticulum. Okay? And there they are going to become membrane bound. And from there, they're then going to pass to what do you call it? Transitional elements. So they're then taken out, and this is energy dependent, let me tell you. Both the transport through the rough endoplasmic reticulum and into the Golgi apparatus, it requires energy. So the elongated proteins, which are now membrane bound, are then passed into the transitional elements, seem to be passed into transitional elements of the Golgi apparatus, your vesicles, and they seem to be pinched off and associated with your Golgi vesicles. Okay, so there seems to be some pinching off of these transitional elements. And after that, they are then transported, taken into the condensing vacuoles of the Golgi mechanism, the apparatus there. Concentrated there, and then passed into zymogen granules, concentrated as zymogen granules. And there, in the zymogen granules, they await for a stimulus. So you have all these proteins. So it's just a synthesis. You require the endoplasmic reticulum, the transport energy, the transport from the endoplasmic reticulum into the Golgi, pinching off, transport condensation or concentration in these condensing vacuoles, and then transformation or conversion into these zymogen granules. And these are what contains the proteins that are released upon a stimulus. Okay, so that is the responsibility of your acinar cells. Okay, and that stimulus starts with the intake of food into the mouth, into the system, cephalic phase. And of course, if you can recall, that is going to depend on acetylcholine. But it's also going to depend on a humoral surplus of hormone, CCK. Okay? All right. We had indicated that the majority of your enzymes that are produced by the acinar cells are inactive precursors. There are just a few that are actually active forms. And these are the alpha amylase, very similar to the salivary amylase or tyline, and they carry out similar functions in splitting your carbohydrates. Pancreatic lipase and antitrypsin, as we referred to before, it has to be active to keep the other inactive precursors inactive. So these are secreted as active forms in the pancreatic secretion, that small volume of proteins. Activation begins in the intestines. That is where it ought to begin, not in the pancreas, in the intestines. And activation of all the enzymes secreted into the duodenum starts with the activation of trypsinogen. We don't want that protein being activated in the pancreas, and that is why we have antitrypsin. So activation occurs in the small intestine by the activation of trypsinogen. And trypsinogen, the active form is trypsin, and that requires an enzyme released from the brush border cells of the small intestine called enterokinase. And enterokinase is also stimulated to be released by CCK, cholecystokinin. So here we see the picture of how important hormones are in the whole process of digestion. So enterokinase is released from brush border cells in the small intestine, and it activates trypsinogen when it's released into the small intestine to trypsin. And once trypsin is formed, it is going to activate chymotrypsinogen to chymotrypsin, and all the others will follow. They will all become activated. Of course, amylase is already active, and most of the pancreatic lipases are already active forms, so they can carry out their digestive process. Yes, question?
This you mentioned earlier that is the reason why one of the cells are inactive. And you mentioned that there's a disease. This is that a person can have it's intentional. So I was wondering what that is.
I said pancreatitis is one of them. Yes, it will cause pancreatitis. But antitrypsin, blockage, gallbladder stones, too much alcohol, these are all things that can cause problems with the pancreas and cause especially blocking stones, anything that block the movement of the isotonic secretion of juice of enzymes out of the pancreas due to some block in the ducts like gallstones that will overwhelm the little antitrypsin and cause activation. So the antitrypsin is there really to keep things normal, as long as there is nothing that is causing the enzymes to even concentrate more and to overwhelm the antitrypsin. Okay, we talk about cystic fibrosis. That is another condition that can cause this problem because it doesn't cause the secretion of but that is at the end of the lecture. Okay, and the pathologist will talk a little bit more about it. But I will refer to that again at the end of the lecture, towards the end. All right? The important thing is to know that the enzymes are normally inactive when it's secreted out, except for those two and the antitrypsin, to ensure that they stay inactive in the pancreas. But once secreted out, we don't longer need antitrypsin. They are going to be activated because of the activation of trypsin, and that requires enterokinase. There are some lipases produced by the pancreas that can be activated via bile salts. These are prolipases to the active form lipase, but the majority of pancreatic lipases are already secreted as active forms. But there are a few bile salt activated lipases too. Okay? Talk more about the other thing that you're. So this is the process of activating our enzymes. Now the neural and humoral regulation. It starts in the head. So you start thinking or condition all that, and just the ingestion of food, right? Is going to stimulate your acetylcholine. And that will account for maybe some 20% of the pancreatic secretion of enzymes, okay? Vagal reflexes are also important, but largely because of stretching of the stomach, yes, which can cause some gastric, which doesn't contribute very much. But certainly, drum release of CCK due to just thinking about the food, you may release some CCK, which is part of the vagal reflex. I'll explain that to you. But acetylcholine is the neurotransmitter that is primarily responsible for the cephalic phase. The gastric phase doesn't account for very much of the pancreatic secretion. Gastrin doesn't seem to have a great deal of effect. Although you learned that gastrin and CCK are very similar, they belong to the same family, they're very similar in action. But gastrin accounts for very little of the pancreatic secretion. And as a short secretion. Here we are, the acetylcholine is actually preparing the acinar cells to release the enzymes. But it's in the intestinal phase that most of the secretion occurs from the pancreas, that enzymatic and isotonic secretion. And this is related to the proteins, fats, and hydrogen ions and other factors in the duodenum because of that chyme that has entered the duodenum. And they're going to stimulate the endocrine cells that are present in the duodenum. So 80% of the pancreatic secretion is as a result of the chyme that enters the intestine. So the intestinal phase of secretion is the main phase of pancreatic secretion. Just to recall, secretin is released from SF due to acidity. And it is, if you look at the diagram, you can see the relationship there. And that secretin, as we pointed out, is going to cause, is the stimulus that causes increased cyclic AMP, which is the second messenger needed to open your cystic fibrosis transmembrane regulator chloride ions, right? Oh dear. All right, you can't say anything. That cyclic AMP, due to the release of secretin, is what is needed to cause opening of the cystic fibrosis transmembrane regulator chloride channels in the apical membrane of the duct cells. Are we going to get it back? And it's going to come back. No light there.
Somebody has to turn back on the projector.
And so that secretin from S cells, then we have, of course, the chyme will contain things like fats and amino proteins, fats and proteins. And these will stimulate I cells. And I cells secrete cholecystokinin. Take a long time, boy. So the I cells, due to the presence of fats and protein, amino acids, will release CCK. Now, when you see the diagram again, you will see that CCK in humans, we have a lot of diagrams from rodents and so we do a lot of experiments, but in humans there are no CCK receptors on the pancreatic acinar cells. But CCK is going to be very important with regards to the release of enzymes. Why? Because it appears that CCK, thank God for Jesus, yes, CCK, look at the diagram very carefully, you will see that CCK, let me see if I can get this mouse. CCK, look at this region, guys. CCK is doing what? It's stimulating the afferent vagal fiber endings. Okay? It stimulates the afferent vagal fibers. And if the afferent vagal fibers are stimulated, they're going to send action potentials up to the higher center, to the center in that brainstem region there, and back down via vagal efferent. So the CCK in humans is more important with regards to the vagovagal reflex. And so you see the vagal efferent coming down, will come down to the acinar cells to cause them to release lots of enzyme secretion. Okay? Then we have other hormones like GIP, gastrointestinal peptide. The right name is really gastric insulinotropic peptide. Okay, insulin dependent. Okay, causes insulin release due to the presence of carbohydrates. Glucagon, these are other hormones that also are released due to the presence of carbohydrates and to some extent fats. And they are going to also be important in, of course, the release of the pancreatic secretions and the digestion digestion, and so on and so forth. Now, this diagram shows you that it's sort of misleading because it's really from a rodent model. It just wants to show the mechanism of action of your neurohumoral substances. Just to point out to you that CCK in humans really acts to stimulate acetylcholine, which then acts on the muscarinic receptors to increase your intracellular calcium. Okay, right? And you also have secretin, which acts on the duct cells largely to increase cyclic AMP, which will cause the bicarbonate secretion. Okay, CCK really acts through that vagal reflex in humans. Okay, so that is just what I wanted to point out in that. So it's cyclic AMP and the intracellular calcium that will increase, and these are the second messenger systems that will either create the CFTRs, chloride channels, in fact open them, increase protein kinase, increase protein synthesis, allow channels to form, and that sort of thing, take care of the mechanisms for both the electrolytic and the enzymatic mechanisms of secretion. It appears that acetylcholine and CCK can also potentiate the effect of secretin. Okay? In other words, cause secretin to be much more active than, it's not just an additive, it's make it much more efficient in its action, creating that large volume of your alkaline bicarbonate secretion. So back to your question, and the pathologist will take care of these things. Pancreatitis is the autolysis of the pancreas. And if the enzymes become activated, if antitrypsin becomes overwhelmed for some problem, maybe due to inflammation of the pancreas, too much alcohol ingestion, gallstone blockage of the ducts so that the secretion can't be released out, then the antitrypsin can be overwhelmed. And that will cause activation. If one molecule of trypsin becomes activated, it's going to activate the other enzymes. And if that happens, then you think that phospholipase A will become activated. Forms lysolecithin, and lysolecithin will damage the cells of the pancreas. And if they are damaged, then of course the pancreas can't work. So blocked pancreatic ducts as well as a damaged pancreas is going to cause serious disease. Acute pancreatitis, chronic pancreatitis, they will tell you more about it. Okay, so you don't want to overwhelm that trypsin inhibitor by blocking the ducts or causing some other problem that can cause problems with the function of the pancreatic gland. Okay? Right? You understand? So it's just that the more you block the ducts or the more is inflammation, things can't pass out, then is the more concentrated the other enzymes will be to the ratio of antitrypsin. And so it can't really take care of keeping them inactive. So once you get activation, one molecule of one, you're going to activate everything. Okay? And then you have Zollinger-Ellison syndrome that you should know about. That is when the delta cells of the pancreas have become tumorous. That's another pathology. Cystic fibrosis, what we mentioned, and the importance of that chloride transport. Cyclic AMP due to secretin. But if there is defect in the production of these channels and the laying down of the channels in the apical membrane to provide for the secretion of bicarbonate, you cannot secrete out that bicarbonate. You're going to have problems with that isotonic secretion. It is going to cause problems with the enzymes that are also being secreted, and so you're going to have problems. Cystic fibrosis. And as I said earlier, the pancreatic exocrine secretions are essential to digestion and absorption. You you are going to have things like that resulting. Yes. Yes. All right, so that is the lecture for us today. So I will upload it for you as soon as I can. Okay, I have other sessions right now, so you have a little reading over the weekend, of course. All right, guys. See you on Monday.